So, there are two big categories of chemical reactions in cells. We have catabolic chemical reactions and anabolic chemical reactions. To review briefly, catabolic chemical reactions are when we take big molecules and we break them down into smaller molecules. This releases energy. Anabolic chemical reactions are when we take smaller molecules and combine them together to make a bigger end product. If we look at catabolism, catabolism is a term that refers to the metabolic chemical reactions that break down molecules. So in our catabolic chemical reactions, we'll use enzymes, and these enzymes are going to regulate these chemical reactions. They will help us take these larger molecules and break them into smaller molecules, which we then later use during anabolic chemical reactions as we mix and match. A good example would be if you took your Legos, two different Lego sets, and you broke them apart into individual Lego blocks, and then you use those individual Lego blocks to make a new spaceship or a new car or a new truck or something new out of Legos. As we look at our catabolic chemical reactions, we'll notice that there's a lot of reducing power. These chemical reactions are going to take high energy electrons and attach them to NADs and FADs, nicotinamides and flavonoid groups. And these high-energy electrons will be attached to these electron carriers that will then shuttle these high-energy electrons around the cell. We're going to see that there's a lot of energy being released during these catabolic chemical reactions. This energy is going to be stored in the form of ATP. Some could argue that this is one of the primary goals of these catabolic chemical reactions, to take energy and dump it into ATP, to make lots of ATP. We need to make large quantities of ATP to power anabolic chemical reactions, and we are going to produce both NADH and FADH2 and ATP during these catabolic chemical reactions. If we look here, we can see during aerobic respiration, so the with oxygen respiration, that there are going to be three big steps. We'll have glycolysis, then the citric acid cycle, also known as the Krebs cycle, followed by the electron transport chain. Now this is with oxygen. So these metabolic pathways are all going to continue and keep going. We'll have a divergent pathway, and here we have a cyclic pathway. This will be a linear pathway down here, and then at the very, very end, we have an oxygen as a terminal electron acceptor, and that oxygen will accept electrons at the very end of this metabolic pathway, and then we make lots of ATP. Note that if we have aerobic respiration, we have 36 to 38 ATP molecules being generated at the end. Quite a few ATPs, and this is a generation of ATP per glucose molecule. So for every one monosaccharide, one glucose molecule that goes in, we can get 36 to 38 ATP molecules at the end. We can also have anaerobic respiration. This is respiration without oxygen. So at the very end, we have a non-O2, a non-oxygenic species. Generally speaking, it's going to be sulfate, nitrate, or carbonate at the very end. So it'll be an inorganic polyatomic ion. That's the final electron acceptor. And depending on which inorganic polyatomic ion we use, you can get as little as 2 ATP at the very end per glucose that goes in, or as many as 36 ATP at the very end per glucose goes in. Aerobic respiration is the gold standard. We will always get the most ATP from aerobic respiration. You can get almost as much from anaerobic respiration, but it is unlikely. And then finally we have fermentative pathways. A fermentative pathway requires no oxygen, and it is not going to use the citric acid cycle or Krebs cycle. It does not have the electron transport chain, but it will have glycolysis to some alternative fermentative pathways. One thing to note is that the fermentative pathways produce hardly any ATP. So because these fermentative pathways produce hardly any ATP, if the microorganism had a choice, it's going to be in the aerobic or anaerobic pathways. It's going to be in the pathway, selectively choose the pathways that make lots of ATP. When we manipulate microorganisms to go down their fermentative pathways, we need to expose those microorganisms to unfavorable environmental conditions so that they go through fermentation and make those wonderful byproducts at the end that are a result of the fermentative pathway. So let's look at aerobic respiration. Respiration with oxygen at the very end. There's going to be a very complex series of chemical reactions that will start with a glucose molecule and at the very end turn it into carbon dioxide. and water. This is a slow burn of the glucose molecule. And by slowly burning or combusting this glucose molecule, one chemical bond at a time, we can harvest and store the maximum amount of energy possible from this glucose molecule. Again, we'll take one glucose molecule at the beginning and store its energy in 36 to 38 ATP. So quite a few ATP. There are three primary steps to aerobic respiration: glycolysis, the Krebs, citric acid cycle, and then the electron transport chain. Aerobic respiration, as its namesake implies, relies on a free oxygen as the final acceptor at the very end of the electron transport chain. Aerobic respiration is done by many bacteria, fungi, protozoa, and animals. We as human beings rely on aerobic respiration. It's the reason we need to inhale oxygen. The moment we stop breathing is the moment that our aerobic respiration rates start to slow down, and if we hold our breath for too long, we'll either pass out or we'll die, depending on whether or not we're voluntarily holding our breath. I digress. We also have anaerobic respiration. So we're still going to have the glycolysis, Krebs cycle, and then electron transport chain, but we don't have a terminal oxygen as a terminal electron acceptor at the very end of the electron transport chain. Instead, we'll have the nitrate, the sulfate, or the carbonate. It's just an oxidized inorganic polyatomic ion as the final electron acceptor. And then finally, we have those fermentative pathways. They do not require oxygen. They are inefficient, and they do not give you a lot of ATP. But a nice byproduct of the fermentative pathways is we get all kinds of exciting and interesting organic molecules at the end, depending on the microorganisms. So we can get our multiple acids, we can get our alcohols, or we can get some vitamins being produced through fermentative pathways. Let's talk about glycolysis. This is the first metabolic pathway, the first of three metabolic pathways during respiration, whether it's aerobic or anaerobic respiration. During glycolysis, we have one glucose molecule going in and two pyruvate molecules going out. One glucose is a chemical formula of C6H12O6. 6 carbons are in glucose, and at the very end, we have two 3-carbon pyruvate molecules. Glycolysis will take place in the cytoplasm in both eukaryotic cells and prokaryotic cells. And glycolysis is used for either aerobic or anaerobic pathways and for fermentative pathways as well. We always are going to start with the glucose, end with the 2 pyruvate molecules, and those pyruvic acid molecules are an intermediate molecule. We can funnel them to the Krebs cycle, the citric acid cycle, or to those other metabolic pathways. Here's a figure from your text, essentially illustrating the same thing. We start off with 1 6-carbon glucose, and we end with 2 3-carbon phosphates. Excuse me, 2 3-carbon pyruvates, not phosphates. Also worth noting is that glycolysis is a net yield of 2 ATP. The net yield of ATP from glycolysis is 2. Glycolysis will yield 2 ATP. The citric acid or Krebs cycle will yield 2 ATPs, and then there's a variable amount of ATP at the very end from the electron transport chain. So, let's say we're on our aerobic or anaerobic respiration, and we're going to use the 3 big metabolic pathways: glycolysis, the Krebs cycle, and then the electron transport chain. If we're going through the 3 big pathways, we'll take that pyruvate molecule and then dump it into the Krebs cycle. Now, to get that pyruvate molecule into the Krebs cycle, we need to change it just a little bit. It's not quite ready to be immediately plugged into the Krebs cycle. We need to take that pyruvic acid and change it so we can go into the Krebs, aka citric acid cycle. We'll change it into a molecule known as acetyl-CoA. So we'll take pyruvic acid and turn it into an acetyl-CoA molecule, and then that acetyl-CoA molecule can be dumped into the Krebs or citric acid cycle. Our Krebs cycle is going to take place in the cytoplasm of prokaryotic cells. And this is where it can be kind of tricky. There's going to be differences now between our eukaryotic cells and our prokaryotic cells. In prokaryotes, we do not have any membrane-bound organelles. So the Krebs cycle just takes place in the cytoplasm of the prokaryotic cell. In eukaryotic cells, we have mitochondria. We do have membrane-bound organelles. So in our eukaryotic cells, the Krebs cycle is going to take on the Krebs cycle happens on the inside of the mitochondria. During the Krebs cycle, we'll take energy from the acetyl-CoA that we're dumping into the molecule, and we'll take that energy stored in the acetyl-CoA and use it to energize high energy electron carriers. We'll take NADs and FADs and turn them into NADHs and FADH2s. So we'll have lots of high energy electrons being carried away from the Krebs cycle. You could think of the overall goal of the Krebs cycle is to take high energy electrons to the next step. The overall goal of glycolysis is to take glucose and to break it down into pyruvic acid. Those pyruvic acids are turned into acetyl-CoA. Acetyl-CoA is fed into the Krebs cycle, and then we rip high energy electrons away from the Krebs cycle and carry them to the next step. A byproduct of the Krebs cycle is going to be carbon dioxide and 2 ATP molecules. The first molecule that acetyl-CoA binds with in the Krebs cycle is oxaloacetic acid, and that turns into citric acid. Acetyl-CoA plus oxaloacetic acid is citric acid. So if you hear me slipping up and calling it citric acid cycle, that's because that is a more current name. The author of this text that you have for our class chose to use Krebs cycle, which is a more dated term for this metabolic pathway. Since Krebs cycle is the term that's used in your textbook, I'm trying preferentially to use that term Krebs cycle. But no, Krebs cycle means the same thing as the citric acid cycle. There are two names for the same metabolic pathway. Here we can see in the Krebs cycle that a pyruvic acid, or pyruvate, is turned into acetyl-CoA. Acetyl-CoA is combined with oxaloacetate to form a citric acid or citrate, and then it's a cyclical cycle that pumps out 2 carbon dioxides and 2 ATPs, and then a bunch of high energy electron carriers, 6 NADHs and 2 FADHs. So when you think Krebs cycle, the main thing I want you to focus on is that we carry lots of high energy electrons away from the Krebs cycle. Where do we send those high energy electrons? We send them to the Electron Transport Chain. The Electron Transport Chain, also known as the respiratory chain, is a series of special carriers that are embedded in a membrane. These series of special carriers are going to need those high energy electrons that are carried by the NADHs and FADH2s. We generate NADH and FADH2s during the glycolysis pathways and the Krebs cycle pathways. During the electron transport chain, those high energy electron carriers are going to drop off their high energy electrons. And those high energy electrons are going to bump from molecule to molecule and will carry their high energy electrons from one molecule to the next molecule. During this process, these high energy electrons are going to lose a little bit of energy every time they bump from molecule to molecule into sequential orderly fashion. As this energy is being lost, it will be used to move hydrogen ions against their concentration gradient. And at the very end of the electron transport chain, we are going to have an electron acceptor. This electron acceptor, generally speaking, is going to be a molecule that is very electronegative. It has a strong affinity for electrons. Oxygen is a fantastic electron acceptor. During our anaerobic pathways, we'll have an SO4, we'll have our sulfates, our carbonates, or our nitrates as those final electron acceptors. During aerobic respiration, if we combine electrons with an oxygen, that oxygen will then recruit hydrogens from the solution and form a water molecule. We have 3 primary proteins. We have 4 primary proteins in our electron transport chain, or 4 primary compounds that are used. I should not say proteins. Some of these are not proteins. We have NADH dehydrogenase, then we'll have a flavoprotein, then we'll have coenzyme Q, and then we'll have our cytochromes. So our electrons will go from one to the next, to the next, to the next. A cytochrome is going to be a molecule that has a very tightly bound metal in the center, and this tightly bound metal ion in the center is going to actively be involved in accepting electrons from the high energy electron carriers and donating them to the next carrier in the series. So here's a figure from your text illustrating this process. Here we have the cell wall of a bacterium. Here we have the cell membrane or cytoplasmic membrane. We'll have an NADH carry its high energy electron and drop it off at the first protein in the electron transport chain. And that high energy electron bounces from protein to protein to protein to protein. As that high energy electron is bouncing from protein to protein to protein to protein, it's going to lose energy, and eventually it will bind with an oxygen molecule at the end and turn into water during aerobic respiration. During anaerobic respiration, those high energy electrons will bind to the nitrates, the sulfates, or the carbonates, and then form other chemical byproducts. During this process of moving the high energy from molecule to molecule to molecule in the electron transport chain, we will take protons from the cytoplasm of the bacterial cell and move those protons to the intermembrane space between the membrane and the cell wall. And we move those protons against their concentration gradient. This builds up a high concentration of protons on one side of the cell membrane and a low concentration of protons on the other side of the cell membrane. This is known as the proton motive force. Those protons will then move with their concentration gradient through an enzyme known as ATP synthase, and this powers ATP synthase to take ATP and take ADP and turn it into ATP. In eukaryotic cells, the electron transport chain is not going to be between the cell wall or in the cell membrane and in between the cell wall, but instead it's going to be on the inside of the mitochondria, on the inner folds of the mitochondria, known as the cristae of the mitochondria. So in a eukaryotic cell, both the Krebs cycle and the electron transport chain take place in the mitochondria. In our prokaryotic cells, the electron transport chain takes place in the outer cell membrane, and the Krebs cycle takes place in the cytoplasm. So as that energy is going to be released from the electron carriers, those high energy electrons move through the electron transport chain, and eventually the protons that are generated, that proton gradient, will funnel protons through ATP synthase. The process of taking ATP synthesis and coupling it with electron transport is known as oxidative phosphorylation. That's kind of a $10 term. We are going to phosphorylate ATP. We are going to take ADP and attach phosphate groups to it, aka phosphorylation. It's oxidative in nature because we are going to be removing electrons during this process. We remove electrons from NADH and FADH, and the removal of electrons from these molecules are used to phosphorylate ADP into ATP. Each NADH can give rise to up to 3 ATPs. Each FADH2 can give rise to up to 2 ATPs from the electron transport chain. The very last step of the electron transport chain is catalyzed by cytochrome AA3, also known as cytochrome oxidase. Cytochrome oxidase. Refer to that as cytochrome oxidase. That is a much easier name to keep track of. This cytochrome oxidase is going to pick up high energy electrons, and it will combine those high energy electrons with half of an oxygen molecule with some protons to form a water molecule at the very end. So yes, bacteria and human cells, prokaryotic and eukaryotic cells, make water molecules. We actually make some of the water that we need to stay alive. Now, if we look at this terminal step, some side reactions of this respiratory chain are going to be incomplete. If we're dealing with half of O2 molecules, this is kind of messy math. Those of you that spend any amount of time in chemistry know that you should not have fractional coefficients in your chemical reactions. So the fact that we have a fractional coefficient right here should tell you that something hinky's going on. Unfortunately, we are going to have incomplete oxidation occur. And as we have these incomplete reductions and oxidations occur, we are going to have some highly reactive oxygen species being produced. Superoxide ions and hydrogen peroxide ions are two byproducts that are produced. These are toxic byproducts. If we're dealing with an aerobe, an organism that can tolerate or live in oxygen, that aerobe is going to produce enzymes that can neutralize the superoxides or the hydrogen peroxides that are produced. Those are superoxide dismutase or catalase. Superoxide dismutase will neutralize the superoxide ion. Catalase will neutralize the hydrogen peroxide ion. If we look at the streptococcal species of bacteria, streptococcal species lack those two specific enzymes, but they still can grow well in the presence of oxygen because they have an alternative enzyme, peroxidase, to handle the highly reactive oxygen species that it could be exposed to. If we look at anaerobic respiration, so respiration without oxygen, instead of having an oxygen as a terminal electron acceptor, we can have the nitrate, the carbonate, or the sulfates. This nitrate will combine with the terminal electron from NADH, and then this chemical reaction is going to be catalyzed by an enzyme known as nitrate reductase. So nitrate reductase, as its name implies, reduces nitrate molecules, and we'll have NO2, H2O, and NAD positive as products at the end of this chemical reaction. Nitrate reductase is going to catalyze the removal of oxygen from nitrate and will give us these products at the very end. This anaerobic chemical reaction is very important because it helps us with denitrification. It helps us take nitrogen and shift it between biologically available and biologically unavailable forms of nitrogen. So this chemical reaction right here is one of the chemical reactions involved with the nitrogen cycle that helps to get nitrogen from the atmosphere and put it into the soil so that it can be used by plants to stimulate growth. Denitrification occurs sometimes when we'll have species of Pseudomonas or Bacillus bacterium take that nitrogen and turn it into nitric oxide or nitrous oxide. Sometimes it can even be turned back into nitrogen gas. So during denitrification reactions, instead of making the nitrogen more biologically available, we make that nitrogen less biologically available. At the very end, if we turn it into N2 gas, that nitrogen is going to be in a form that is very chemically inert and very difficult for microorganisms and large macroorganisms to use. Denitrification, as I mentioned, is very important for that nitrogen cycle or recycling nitrogen in the biosphere. We'll have other oxygen-containing nutrients being reduced by other bacteria, like those carbonates and sulfates, and those are part of the carbon cycle or the sulfur cycle. But nitrate is the gold standard for aerobic respiration. Nitrate produces more oxidation of nitrate, reduction of nitrate produces more ATP than the oxidation or reduction of sulfate or carbonate. So if we look at terminal electron acceptors, oxygen is the best terminal electron acceptor, followed by nitrate, and then sulfates and carbonates are the worst terminal electron acceptors. Sometimes, instead of going through the Krebs cycle and the electron transport chain, we'll go through some alternative metabolic pathways. These are known as the fermentative pathways. These fermentative pathways are not very efficient. We don't get a lot of ATP from these fermentative pathways. The big benefit of these fermentative pathways is they can always happen in the absence of oxygen. Oxygen is never going to be required. And we can get some very interesting chemical byproducts from these fermentative pathways. These fermentative pathways typically are going to be turned on and off. Most microorganisms prefer not to use fermentative pathways because they don't get as much cellular energy. They don't get as much ATP from the fermentative pathways. But they can activate the fermentative pathway if they absolutely need to, if they are in an oxygen-poor environment, or if they don't have anything else they can use as a terminal electron acceptor in the electron transport chain. So here we can see pyruvic acid. Instead of funneling it down the citric acid cycle, we'll take that pyruvic acid and funnel it down a fermentative pathway. This particular fermentative pathway is the exact fermentative pathway that occurs in human beings. Pyruvic acid is turned to acetaldehyde, which is then turned to ethyl alcohol. Or we can take that pyruvic acid and turn it directly into lactic acid. So pyruvic acid to lactic acid is the one that occurs in human beings. This other fermentative pathway, where we go from pyruvic acid to ethyl alcohol, is an alternative fermentative pathway that does not occur in human cells, but can occur in many prokaryotic cells. Since we take one glucose molecule and turn it into two pyruvic acids, this fermentative pathway is going to happen twice per glucose molecule that enters into glycolysis. So for human beings that are on the fermentative pathway, one glucose will get turned into 2 lactic acids. If we look at a yeast cell that's on a fermentative pathway, that one glucose will get turned into 2 ethanol molecules, or 2 ethyl alcohols. Some of the things that we can make from the fermentative pathways, or that microorganisms can make from the fermentative pathways, include ethanol, carbon dioxide, acetone, butanol, lactic acid, acetic acid, a wide variety of vitamins, antibiotics, and hormones. And if we're looking at the large-scale industrial production of these chemical compounds, generally speaking, we'll take these microorganisms and put them in a chemostat, a device that maintains constant extracellular environments. And those chemostats, generally speaking, will force the microorganisms down the fermentative pathways so that we can have large vats producing large amounts of these chemicals that we can use as building blocks for more complex molecules. Sometimes we will have catabolism occur from non-carbohydrates. So up until this point, we've been talking about taking glucose and breaking it down to make lots of ATP. But we can also get energy from lipids or from proteins. Glucose is not the only energy-yielding macronutrient or biological compound. If you look at your calorie information on the food that you eat, on the label, you'll notice that there's calories in protein and that there's also a lot more calories in lipids or fats. If we take a complex polysaccharide, like starch or glycogen, we need to break that complex polysaccharide down into individual glucose molecules that can go into the glycolysis metabolic pathway. If we have a lipid that's been consumed, that lipid needs to be broken down by a series of enzymes known as lipases, an enzyme that breaks apart lipids. If we break apart a typical fat molecule, that typical fat or stereotypical fat will have 1 glycerol molecule and 3 fatty acids. Glycerol can be funneled directly into the glycolysis pathway. Fatty acids are broken apart 2 carbon atoms at a time and funneled into the Krebs cycle as acetyl-CoA molecules. If we take a protein and consume a protein as a source of energy, we will break down that protein into its individual building blocks, its individual amino acids, with a series of enzymes known as proteases, enzymes that break apart proteins, a protease. Those individual amino acids are then going to have their amino groups, the NH2 groups, are removed through a process known as deamination. The remaining carbon-containing compounds are going to be pumped into the Krebs cycle. Depending on the amino acid, we will pump those leftover carbon compounds into the Krebs cycle at different points of the Krebs cycle. So, concept check class. What is the maximum amount of ATP that we can get per glucose molecule for each of the following types of respiration? This was covered several slides ago, so you can rewind the video, flip back in your PowerPoint, or check your textbook. With aerobic respiration, the maximum yield of ATP is 36 to 38. With anaerobic respiration, the maximum yield of ATP is 2 ATP all the way up to 36 ATP, depending on what that terminal electron acceptor is. And then finally, with the fermentative pathways, the maximum net yield of ATP is 2 ATP. So this has been arranged in order from producing the most ATP to producing the least ATP. If you have any questions about these metabolic pathways, that is very understandable. As an aside, I remember when I was a student and I was first learning about cellular respiration and these metabolic pathways, I found them particularly confusing. So, if you have any questions, post them on the discussion board, shoot me an email, or watch some of the videos that I have made available. Happy studies!